the chromosomal basis of inheritance. We've talked about genetics on a single gene basis, how what happens if you get one copy of one gene and one copy of another gene based on whatever pattern of inheritance that specific gene follows. But there is a much much more overarching idea when it comes to genetics, and that is with chromosomes. So, we are going to start off. I'm going to connect some of our understanding of chromosomal behavior in meiosis to Mendel's laws of inheritance. And then I'm going to kind of talk you through the scientific discovery that was, oh, all of Mendel's rules actually do have like a physical basis. And that starts us off with the fact that Mendel's laws can be explained with chromosomal behavior. So Mendel had two laws, the law of segregation and the law of independent assortment. And both of them, both of those laws have are the way that they are because of the way chromosomes move during meiosis. So in the law of segregation, the two alleles for each gene separate during gamete formation. So the two genes are separated completely, just like Mendel said. And when we are undergoing meiosis, we have completely different alleles on different chromosomes, right? We've got one chromosome or one allele on one homologous chromosome and one allele on another homologous chromosome. They're not together, they are two separate entities. So, our understanding of homologous chromosomes gave us that one. The law of independent assortment, so alleles of homologous chromosomes assort independently during gamete formation, meaning they are, it doesn't, goodness gracious, where are my words today? It's completely random which allele or which daughter cell gets which allele from meiosis, right? It is completely random whether or not a gamete gets the allele for purple flowers or tall stems or pink flowers and short stems or yellow seeds and pink flowers. They are different because they are assorted independently on our homologous chromosomes during meiosis. So whenever Gregor Mendel came up with these laws, it was an abstract concept. He didn't know about how meiosis worked. He didn't know about what DNA was. He just knew that there were some sort of hereditary factors and they followed certain laws of probability. But our understanding of chromosomes, our ability to track certain genes on chromosomes, to look under a microscope and see a cell in meiosis and count where those chromosomes are, is completely supportive of what Mendel thought was going to happen, which kind of goes farther to prove that Mendel's studies were way ahead of his time. So, questions here? Everybody understand how our understanding of meiosis supports Mendel? All right. Cool. So that's that one. We also have the chromosomal theory of inheritance. So this is the actual scientific theory that went to prove Mendel's hereditary units. So the chromosomal theory of inheritance states that genes are located on specific positions on chromosomes, and that this should be that the behavior of chromosomes during meiosis accounts for inheritance patterns. So not only do the way chromosomes move during meiosis give us our law of independent assortment and law of segregation, but also there are specific positions on chromosomes where certain genes lie. So if we were to look at my chromosome 13 and your chromosome 13, whatever genes were on whichever spot on those genes would be the same on my chromosome 13 as your chromosome 13. I don't know what genes are on chromosome 13, or I'll give you a specific example. So we were able to work out the process of meiosis in the 1890s. We had mitosis a couple years before, but it's 1890, and we were able to finally draw parallels from Mendel's work to our chromosomes behavior in meiosis. So it wasn't until we got the ability to see chromosomes condense and gametes form that we were able to say, oh, not only was Mendel right, Mendel was like right. I also have the word loci in here. So a gene's specific position on a chromosome is called its locus. The plural of locus is loci. So if you ever see that, it just means where at location, but scientists like to have a lot of names for stuff. So a gene's locus is just where it is on a chromosome. So this chromosomal theory of inheritance was worked out by a bunch of scientists, but one has the credit for it. Let's get this down before I move on now. Like I said, it's 1890. Mitosis was worked out, I believe, in the 1880s, and then early 1890s we got even farther into our understanding of how cells split up. So this is Thomas Hunt Morgan. He was an experimental embryologist in the early 1900s at Columbia, and his experiments were the ones that showed us that Mendelian inheritance has a physical basis. So that first slide was essentially all thanks to Thomas Hunt Morgan. I'm going to show you how he did it. We're going to talk a little bit about what organism that he used to find this out. It's actually really cool. So yeah, it was this dude right here in the early 1900s that kind of put things into place. And if you think about timeline wise, if we figured out that chromosomes were the basis of Mendelian inheritance in the early 1900s, it wasn't but 40 to 50 years later that we finally worked out the structure of DNA. Just kind of put you in a timeline. So there's a big jump between when Mendel was doing his work and when Thomas Hunt Morgan was doing his work. So we're 1900s at Columbia. Then you jump to England in 1950, and that's when Rosalind Franklin was working with X-ray crystallography. So that gives you kind of the pace at which the world learned about genetics. Yeah. No. He had the mind of a scientist, but the means of someone else. So he didn't have the ability to like go off and study. His family was really, really poor. So he became a monk and dedicated his life to religion. But he knew that religion wasn't necessarily his calling. So he spent most of his time working in a garden with these pea plants and coming up with this pattern of inheritance. So while he didn't necessarily credit himself as a scientist, he had a very scientific mind. So that's Thomas Hunt Morgan. He used a special experimental organism that is still used to study genetics today. So this is a fruit fly, also known as Drosophila melanogaster. If you do any work with genetics, you will run into Drosophila full stop. And his choice in Drosophila for his experimental organism was really, really important. First things first, a single mating pair will produce hundreds of offspring. So no matter what happens, he will always have access to their babies. And it's really easy to see percentages in large numbers. So if he was tracking the frequency that an allele showed up, being able to have the specific percentages in a large population is easier to obtain. Drosophila also only have 4 chromosomes. So if you were to look at a Drosophila cell under a microscope, it is very easy to differentiate between their 4 chromosomes. And within those chromosomes, we have 3 autosomal pairs and 1 pair of sex chromosomes. And their sex determination is identical to humans, where if they have 2 X chromosomes, they are biologically female, and an X and a Y chromosome, they are biologically male, which is exactly how human inheritance works. We'll talk a lot more about sex chromosomes and sex-linked inheritance tomorrow. But yes, so only 4 chromosomes, easy sex determination, and a lot of offspring are just a few of the reasons why his experimental organism was important. They're also really cheap. So keeping fruit flies around is a lot easier than keeping lab mice around, right? It's a lot cleaner. And as long as you don't break a vial, they're easy to obtain. The genetics lab at Purdue works a lot with Drosophila, and you can always tell when the sophomores get into their genetics unit because there are always fruit flies buzzing around because somebody inevitably drops a vial. So, yeah, if you ever in a biology building and you see a bunch of fruit flies around, it's not because it's dirty, it's because they're used as experimental organisms a lot. Wild type versus mutants. So Thomas Hunt Morgan was looking for some sort of easily distinguishable quality that he could study. And he bred flies for 2 years in order to try to find one of these easily determined genes to work on. And after 2 years, he finally found his naturally occurring mutant phenotype. So normally, so a phenotype that is most commonly observed in a natural population is called the wild type. So if a plant is normally pink, but there's some sort of mutation that makes it purple, the wild type would be pink and the mutant would be purple. Traits that are alternatives to wild type are called mutant phenotypes. So not naturally if you were to pick up any fruit fly out of a population, it would most likely have red eyes. That red eyes is the wild phenotype. But eventually, Thomas Hunt Morgan found a fly with a white eye phenotype. So it is easy to see if a fruit fly has red eyes or white eyes, so it's easy to track in a population. So he used this white eye phenotype to carry out the rest of his genetic work. So a wild type has the most commonly observed phenotype, and in fruit flies, that wild type is red eyes, and the mutant is white eyes. So this is within any organism. The most commonly phenotype is the wild type, and the mutant is called mutant phenotype. So anything that's not type. Trying to think of another example. I'll let you know. So, any questions about wild type versus mutants, red eyes versus white eyes? So wild type notation, I figured I'd pop this in here. Probably could have made the font a lot bigger. I will fix that. So the allele for white eyes in Drosophila is a W, and a superscript plus identifies a wild type. So if an allele is, or if somebody's, or if a genotype is W plus, they are a homozygous wild type. If they are W plus W, that would make them heterozygous. If the wild type is dominant, they would have the wild phenotype, but if the mutant is dominant, they would have a mutant phenotype. So instead of like big R, little r, they do WW plus. So if you see that out there, that's why. So this was his experiment. So Morgan muted a mutant male with a wild type female. So a female with white eyes, female with red eyes, and a male with white eyes. Excuse me. And all of their offspring had red eyes. Does this tell us that our wild or mutant type is dominant or recessive? Recessive, because if you cross two together and all of the offspring have one phenotype, we have a F1 generation of all of our wilds. So our F2 generation, when he bred these all red eyes together, that white eye trait popped back up again. And he noticed this 3-to-1 ratio where we got 3 red eyes to 1 white eyes, which is exactly the ratio of recessive inheritance. But the kicker here was that only the males had white eyes. There were no females in the population with white eyes, which Morgan determined that this white eye mutant must be located on the X chromosome, because there was no way for there was no female offspring to have a recessive phenotype, because there was no way that they had 2 copies of the gene. I'll talk a lot more about sex-linked inheritance tomorrow, but from his experimentation, we can, goodness gracious, determine that this mutation is on the X chromosome, and it supports the chromosomal theory of inheritance. So if we've got our homozygous mother and our heterozygous father, we've got our cross here in our F1 generation, getting all heterozygotes. And if we cross a heterozygote together, we will have our 3-to-1 ratio. Let's see how that happens. Okay, so that is how we, this experiment right here is how we got to the point where, the way chromosomes behave actually support Mendel's theory of genetics, using his Punnett squares and showing how chromosomes behave during meiosis. So, questions, comments, concerns? I think the next slide. Yes. So tomorrow we'll talk about sex-linked inheritance a little bit more. Get to talk about calico cats a little bit. It'll be a fun time. But now you have the time to finish up what we did on Friday. If you've already done that, then you have time to chill. If you have any questions, just let me know. Cool.